Good morning, Paras sir.
Yes, ma'am. Just a
Now
minute. We will
you are
start
the
now.
session coordinator for this session. Please take over the charge and start the session
Yes, ma'am.
exactly at 11 a.m. Our invited talk guest, Dr. Mahan Tiwari, also join in the conference. Okay, sir. Over to you. Now you are the session coordinator for this session. Please take over the charge and start the session exactly at 11 a.m. Our invited talk guest, Dr. Mahan Tiwari, also join the conference. Okay, sir. Over to you. You are the session coordinator for this session. Please take over the charge and start the session exactly at 11 a.m. Our invited talk guest, Dr. Mahan Tiwari,
उसमें तो लगा के पहले देखना चालू हो रहा है तो, नहीं तो छोड़ देना, उल्टा सीधा मत लगाना, ठीक है।
It's track 2. Track 2.
Track 2 session chair, track 2 11 a.m.
हाँ हाँ, यस सर, आपका इनवाइट सेशन चेयर अभी से है.
अच्छा, ठीक है, समझ गया. ओके.
ओके, वेट.
Track 2.
अभी भी टर्न ऑफ कर. परमार सर, प्रॉब्लम कर रहा है, बार-बार प्राइवेसी इनका हो जा रहा है. ये देखिए ना, कैमरा's privacy switch remove anything that is covering इधर तो दिया है, पर ये ऑन कैमरा बार-बार करने पे वही चीज आ रही है. पारस सर, प्लीज म्यूट योर माइक. पारस सर. पारस सर.
Yes.
हेलो विमलेश सर. विमलेश सर, कमलेश सोनी सर वेट कर रहे हैं मीटिंग लिंक में ज्वाइन होने के लिए. हेलो अनिकेत सर. अनिकेत सर. Yes, Paras sir, please continue the session. Our invited talk guest also joined with us.
Hello.
Hello, good morning, sir.
Good morning, ma'am. Good morning.
Welcome to the conference link.
Uh,
Paras sir, just a minute. We are starting with our session coordinator. Paras sir will take over the session. Hello, good morning to all of you. I will be starting the session. We are in this track 2 session, and this is the technical presentation of day 2 of our international conference. And now, in this conference, we have technical presentations, poster presentations, and for that, we are having our session chair, Dr. Mahen Tiwari. At first of this technical presentation session, we are having invited talk from Dr. Kamlesh Soni. So I welcome Kamlesh Soni sir. It is pleasure to introduce Dr. Kamlesh Soni as an assistant professor of biotechnology at EKS University, who earned his PhD in genetics from the University of Delhi. His research expertise refined during a postdoctoral fellowship at Academia Sinica, Taiwan, focused on gene regulation, host pathogen interaction, and CRISPR-Cas-mediated crop improvement. Dr. Soni has authored numerous high-impact publications in journals such as the Journal of Experimental Botany and Plant Cell Reports, a multi-layer NET qualifier and recipient of the Young Scientist Medal. He is a recognized leader in biology. Yes, sir. Okay, sir. मैंने start कर दिया है session. And also introduce our invited guest. So I would like to welcome Kamlesh Soni sir for his presentation. Paras sir, please mute yourself. We are starting with the session of Dr. Kamlesh.
I will start. Sorry for the disturbance.
No, sir. Dr. Kamlesh Soni will start the session from his presentation. Over to you, Kamlesh Soni sir.
First of all, good morning to all of you. My slide is visible to all?
Yes, sir. It is visible. Yes, sir.
Okay. So, thank you for providing this opportunity to present the talk. So, basically, my talk is on the brief introduction of my talk is like I would like to explain about the taptum-specific promoter from the plant because the plants are basically taptum-specific promoter used for the expression of the gene which is responsible for the encoding the barnase, that is a non-specific RNase. So if we express any gene, any toxic gene in the tapetum, it will kill the tapetum. There will be no pollen, so the plant totally will be a female plant, right? So in this presentation, we will talk about the taptum-specific promoter that has already been used for development of the hybrid seed technology. But interesting part, this promoter was identified in 1990s, but nobody has looked about its regulation, right? Regulation means why this promoter is so tightly regulated in the tapetum specific, why there is no expression in the leaf, why there is no expression in the root, only in the tapetum layer it expresses. So in this talk, we would like to talk about its regulation part, like how this promoter is regulated, how the gene is tightly regulated in the tapetum tissue specific manner. So in this talk, this is the brief, brief of the tapetum specific promoter. Normally in the wild type, what happens? Just pointer. Right. So in the wild type, you can see there is a normal development, and this is the hello.
You continue, Doctor. You continue.
Okay. Yeah. So this is the normal tapetum development where you can see T for the tapetum, and this is the microspores, right? In the male sterline, what happens? There is no tapetum layer. So there is no tapetum layer, though there is no microspore, because the tapetum is a nourishing tissue which provides the nourishment to the developing microspore, which ultimately develop to the pollens, right? But when and how it is possible when we use this TA29 promoter with the Barnase gene? The Barnase is basically non-specific RNase. It kills all the RNA from the tapetum layer, so there is no tapetum formation, right? But when we use the Barnase along with the bar-star gene, the bar-star is nothing but the inhibitor of the Barnase gene. So when Barnase and bar-star is make a complex, this Barnase gene is inactive, and there is a clear tapetum formation and male microspore development, because this RNase is not active, because Barnase is bar-star is making a complex with this gene, right? So how we use this technology for the hybrid seed production. What we do, we kill the male, and we develop the female plant, and we take another plant as a male, and we put the pollen from the female plant to make the hybrid seed in order to avoid the self-pollination, right? So why tapetum specific promoter? Is to expand the Barnase bar-star technology. There is a need of the tapetum specific promoter, and also to we understand the regulation of this tapetum specific promoter, right? So here it is the just outline of the functional characterization promoter was identified in 1990s by the Coltuno, and he identified there is which region is actually important for the tapetum specific specificity. So what he did, he took the 1.5 kb base pair length, and he started cutting from the 5' end, and he figure out which region is actually important for this tapetum specificity, tissue specificity. But after that, nobody has looked out which actually transcription factor binds to it and regulate its specificity. In this case, we attempt to identify the regulatory elements, where we took the 860 base pair upstream to the ATG size, and we identified there are some transcription factor, there are some cis elements and transcription factor which bind to this promoter. So what we did, we used the two strategy, a loss of function analysis and gain of function analysis. In the loss of function analysis, what we do, we mutate those identified cis element from the promoter region and see what happens, because we lost the function of the promoter. In the case of gain of function analysis, what we can do, we can take the TA29 promoter, fuse with the GUS gene, a reporter gene, and we overexpress the identified transcription factor, so that the transcription factor is overexpressed. Obviously, it will bind to the promoter, and it will express any other tissue also, because the transcription factor is the key regulator of the promoter, and transcription factor if bind to the promoter, and if we enhance the transcription factor activity by the 35S promoter, it will express everywhere. So it will bind to the promoter everywhere. So it will start expressing the tapetum, this TA29 everywhere, because it was restricted to the tapetum. Now it will start to other tissues also. Right? So this is how we have identified the genome sequence. We have did the cloning. In the case of loss of function analysis, what we found in the anther tissue, you can see the LGD gene was found to be expressed when the promoter is lost. This is the negative control we have used. So we found out of the transcription factor we identified, MIP12 was identified as a positive regulator, because in the case of wild type, there is expression is like 3000, But when this site is mutated, MIP12 site is mutated, the transcription factor, the promoter activity is downregulated. That means this element is very important to active in the tapetum. In the case of GT1A and NAG1A, there is the same situation, but interesting observation was found in the case of LJJ when activate, when the elements, the promoter elements is mutated, activity enhanced. That means this is the elements which is actually a negative regulator, because the when we mutate this site, promoter activity is enhanced. Not only enhanced, it also start expressing in the other tissue than the tapetum. Right. So this is again the same figure for the total protein in the different tissues. So this is the loss of function analysis, the statistical analysis, and we found MIP12, GT1, and NAG1 site binding site is a positive regulator, and the LZHG binding site is a negative regulator. So gain of function analysis, again I have explained that we will take the transcription factor overexpress and it will bind to the TA29 promoter, and if it bind, it will definitely express everywhere, and we found the promoter is actually the GT1 and MIB1 and NAG1 is actually the regulating the this promoter in a positive manner. So interesting observation, this negative regulator is also found to at the ectopic level. Ectopic means other than the tapetum specific area. When we mutate this transcription, we mutate this cis element, the promoters start expressing other than tapetum also. You can see in the leaf, in the calculus, in the shoot. From observation. The binding elements for the MIP, GT1 and NAG1 are the positive regulator, as mutating them result in reduced activity of the TA29 activity in the anther. If the mutate activity is lost and promoter become less active. But LGDG was found to be a negative regulator,
Sir, I
because
think your
once the activity,
connectivity is low.
once the
Please come in.
is hello. Yeah. So activity is increased, not only increased, but also start giving in other tissues also. So this is the direct interaction. Actually, this is the work done for the cis element only, but we have to identify exactly which elements in these transcription factor bind, and that is only possible via the protein-protein interaction analysis, that is electrophoretic mobility shift assay or the Y2H assay, Y1HA need to be done to make it more strong data. Thank you. I'm done.
Coordinator, Parasji. Hello.
Formatively enriched our lesson. At that time, my connection was not good.
Please continue. Invite the second speaker.
He has come from Taiwan and he has a mastery of technology. I think our student has enriched very much from his. Next, first presentation is from Varsha Singh, BTech 8th semester Biotechnology, and her topic is assessment of antimicrobial properties of endophytic EMS1 against Fusarium oxyporum and human pathogens. I invite Varsha Singh to present her topic. Hello, Varsha, have you joined meeting? Hello.
Varsha, please unmute yourself and continue presentation. Okay. Is there any technical issue?
Varsha? Please reply if you are ready or not. Otherwise, we will start our next presentation. If Sakshi is ready, she may start the presentation.
Sir, good morning.
Yes. Good morning.
Sir, firstly दिव्या दीदी का करवा लीजिए.
Yes.
दिव्या गौतम का सर करवा. She is ready, sir.
Next, Sakshi Misra और संजना सोनी और नेहा पटेल. Hello. Hello. Sanjana. Hello. Hello.
Professor Parasji?
Yes, sir.
Please call the second.
The students are not ready.
हेलो सर, पारस सर जो प्रेजेंट है उनका नाम
Yes.
आप कॉल कीजिए ना.
वही बोला,
तब तक बाकी
प्लीज
लोगों
कॉल
को कॉल
सेकंड.
कीजिएगा. जो प्रेजेंट है उसको कॉल
Yes,
कीजिए.
मैम. Yes, मैम, उन्हीं को कॉल कर रहे हैं अभी. इनका तैयार.
ठीक है, कोई दिक्कत नहीं. जो सेकंड हेड है उनको कॉल करके उनको करवाइए.
Yes, sir. If poster presentation participants are ready, then they
हेलो,
will start.
सृष्टि सोनी.
Meanwhile, they.
Srishti Soni, are you there?
Yes, ma'am. Yes, ma'am.
Are you ready with your PowerPoint presentation?
Yes, ma'am.
Okay, start with your presentation. Share your screen.
Yes, ma'am. 1 minute, please.
हाँ.
पीछे थोड़ा.
Is my screen visible?
Yeah, visual.
Srishti, please auto rotate on कीजिए अपने फोन का and pick your phone in horizontal mode. Auto rotate on कीजिए और मोबाइल को horizontal mode पे रखिए. Yes,
My
yes.
PPT is visible.
Good, beta, good. Continue.
Okay, ma'am. Good afternoon, dear faculties. Myself, Srishti Soni. I'm today presenting my presentation on bioremediation enhancement of hydrocarbon contaminated site by using indigenous bacterial strain. Let us move towards the introduction. Pollution with crude oil and its products has become globally a major environmental concern. Human activity has led to release of the liquid petroleum hydrocarbon into environment, causing the pollution of marine coastal water, shorelines, and land as well.
Beta,
The
speak
environment near the
loudly.
petrol,
Speak loudly,
the
beta.
environment near the environment near a petroleum refinery or a tanker terminal can be freely exposed to the chronic oil pollution from the frequent spills. So, area of the research may be one of the method of cleaning up oil spills that has been explored in the use of oil-eating or oil-loving microbes, a process known as bioremediation. Several bacteria species belong to Pseudomonas, Stenotropomonas, Rhodococcus, Brevibacterium. Those are the genera that have been reported to be degrade hydrocarbons. Whereas there are four objectives we have worked on: to isolate and identify oil degrading bacterial strain from contaminated environment, to evaluate the hydrocarbon degradation potential of the selected strain in free and the immobilized form, to compare and biodegradation of efficiency of the free and the immobilized bacterial culture, to analyze the emulsification index by the biosurfactant product. Let us move towards the methodology. The first objective we have worked on: to isolate and identify oil degrading bacterial strain from contaminated environment. The first oil sample we have collected from an oil factory where the oil was being refined, produced and refined. The nearby area was hardly contaminated, heavy contaminated with an oil. The oil was preserved in the laboratory condition from which we have performed serial dilution, and bacteria were grown in the cultural plate, whereas mixed culture were obtained the slant, and they were stored in the further culture plates, and the pure culture was attained from those plates. Whereas we have measured the bacterial growth in minimal salt medium by preparing two flasks in which MSM was at the 50 ml concentration. One was as a control with oil, and other was with 1 ml of an oil. Both of them were cultured with the same bacterial strain S1 and incubated for the 48 hours at the room temperature. Whereas measurement of bacteria with the nutrient broth media was taken place with two flasks taken 50 ml of nutrient broth media in both of the flasks. One was with 1 ml oil and one was as a control with no oil. Both of them inoculated with the same bacterial strain S1 and incubated for the 48 hours, whereas their optimal density was taken at every 30 minutes. Whereas let us move towards next objective: to evaluate the hydrocarbon degradation potential of selected strain in free and immobilized form. Oil bioremediation experiment was performed in which 5 different flasks were taken. All of them consist of MSM media at the 50 ml concentration, and all of them were having an oil at the different concentration from 1 ml to the 5 ml. From increasing level, all of them were inoculated with the bacterial strain S1, which we have incubated, whereas all of their optimal density was measured from first day to the third day, fifth day, 7th day, and 10th day. Whereas they were incubated at the room temperature, and at last all of their oil drop test was performed. Let us move towards oil drop test procedure. The ELISA plate was taken, consists of 96 wells, will be used to perform oil drop collapse test assay. The well was named as I1, I2, I3, I4, I5P, and I6N, whereas I5P is a positive and I6N was a negative control. I1 and I3 control wells, I2 and I4 were as test wells. Whereas in next to test of ELISA test, the wells will be named as I1, I2, I3, I4, I5, and I6P and I7P. Whereas P was positive and N was as negative control. It the 2 microliter of oil into the center of each and every wells to be tested, and IP will have the 5 microliter of SDS, and I7N have 5 microliter of water as negative control. Let us move for the immobilized bead preparation. We will dissolve 0.3 gram of sodium alginate in 10 ml, stir it till the homogenized mixture was obtained. 3 to 4% solution was prepared. Then let the solution to be stirred for 30 minutes and remove the air bubble. Now we will mix the 1 ml of culture broth in this. So 3% to 4% of the sodium alginate solution. We will load the solution in the syringe and drop the solution into the axis 100 ml of a stirrer. 0.4 to 0.5 ml of the calcium chloride solution. CaCl2 solution. The beads are required to leave the calcium solution to cure for 3 hours. After that, we will clean the beads and can use them for the experiment. All bioremediation experiment using immobilized beads. Different 5 flasks were prepared. All then consist of MSM media of 50-50 ml, whereas all of them consisting of oil from 1 ml to the 5 ml in increasing concentration, with the beads added to them in an equal amount. All of them were subjected to taking the optimal density from first day, third day, fifth day, seventh day, and 10th day, whereas the incubation at the room temperature was taken. Let us move towards the biosurfactant production. LB media was prepared with the 2 ml oil in it, and its pH was adjusted. Then we have added the 10% of the culture media inoculated in the broth. We'll incubate them at the 30 to 37 degrees Celsius for 72 to 120 hours at the 150 to 180 RPM. Centrifuge was done of the fermented broth at 10,000 RPM for 15 minutes. We will acidify the supernatant at pH 2 using 6N HCl and refrigerate overnight. Next day, we will centrifuge the same broth and collect the precipitated biosurfactant, and will mix the 2 ml of biosurfactant with 2 ml of oil. Will vortex in 2 minutes. Then we will measure the emulsion height after 24 hours and calculate the emulsification index by using our formula. Let us move towards the final result. The gram staining we performed, it shows that the isolated bacterial strain were pink in color, and the pink in color of the counter strain indicate that the bacterial strain is gram negative in nature. Appearance was cocci. The growth with the MSM media, the strain S1 showed the higher OD have compared to the control, indicate the better initial result in the lag phase. After the OD of the strain 1 increased, it keeps on increasing and then attained the peak. After about the 30 minutes, showed the enhanced growth in the presence of oil from lag to log phase. Whereas after 30 minutes, the OD of the strain S1 gradually declines and then stabilizes, possibly due to nutrition depletion or onset of an stationary phase. Whereas in contrast, the control culture without oil showed the minimal change in OD during the first 60 minutes, but display a gradual increase, therefore, approximately at 120 minutes. As we have measured the bacterial growth in the nutrient broth media, growth was measured at the optimal density at 660 nanometer. Control sample showed a higher OD value compared to an strain S1, indicating the better initial growth in the control sample. About 30 minutes later, the OD of this strain S1 attains the peak from lag to the log phase compared to the control sample. In the next 90 minutes, the OD of the strain S1 gradually declines and then stabilizes. In contrast, the control culture without the oil showed the minimal change in OD during the first 60 minutes, but displayed. Let us move towards bacterial growth curve of the oil bioremediation experiment. As we have performed the experiment with the 5 different flasks at the 5 different oil concentration, the graph shows that the maximum percentage of oil degradation was found in 1 ml of crude oil concentration in MSM media. The percentage of oil degradation decreases at the concentration of oil increases in the MSM media. At the 5 ml oil concentration, the degradation of oil was least, is most likely due to toxicity of the oil on the tested isolates. After the 10 days later, the optimal density recorded for the 1 ml oil drops significantly. The quantity of the oil degradation is not same as it was from the starting day. Oil drop test was performed for 2 different experiments. It shows that the I1 was showing the diffused edge, I2 was showing the irregular and rough edge, whereas I1 and I3 were controlled as the test remain intact and show no collapse of the oil drop test. And the other, for the bioremediation experiment oil drop test we performed, it shows that I1 show irregular edges and I2 shows diffused edges. I3 had the rough and the diffuse edges, exhibits the complete oil drop collapse. I4 had the crescent shape and I5 showed the round shape and intact edges. This did not reading of the collapse of oil drop indicates the negative outcome. Significance of oil drop test: low cost and simple quantity assay, positive result used in the initial selection of potential strain for bioremediation. Bacterial growth curve for oil bioremediation experiment using immobilized beads. Beads are introduced which have been made of the alginate. It will enhance the process of the physical support. The maximum percentage of oil degradation was found at the 1 ml of the oil concentration in MSM medium. The percentage of oil degradation decreases as the oil concentration straight increases. At the 3 ml and the 5 ml of oil concentration, the degradation of oil was less compared to 1 ml and 2 ml of oil MSM medium. At the time interval of 10 days, the 1 ml oil, there is a significant drop in the growth curve, with from stationary to the death phase is likely due to the oil degradation in this is not same as it was in the static days. Significance of immobilized beads: facilitate gradual and sustained microbial activity, protect microorganisms from environmental stress, improve longevity. We have compared the bioremediation efficiency of the free versus immobilized bacterial strain. Both of the graphs were shown as the comparison, in which the growth curve of an immobilized bead showed the better growth
Please,
compared to the
Srishti,
free bacterial curve.
please
Yes.
conclude. Please conclude. Your time is going to over.
Yes, sir. Yes, sir. And in the 1 ml shows the highest growth in both conditions, but the one that beat achieve have the highest peak rate. To analyze the emulsification index of the biosurfactant production, both of the we have observed the emulsification index in the 72 hour and 120 hours. The 72 hour shows the better emulsification index of 40% as compared to the 120 hours, because of the bacterial death for the 120 hours of incubation. Significance of biosurfactant: enhance oil dispersion in the contrast site and promotes the faster microbial degradation of hydrocarbons. Let us move towards the conclusion. The increase in the environmental pollution with the oil spread being a greater contribute has made it necessary to find a way of the eliminating the harmful effective effects on human and the ecosystem. Discussion last will be moving towards the discussion. The beads are more supportive to the bacterial growth as it protects them from unwanted contact with the foreign materials. Oil degrading capacity of the free culture is observed but is not better than the immobilized beads of the bacterial strain. Last but not the least, references. Thank you.
Thank you, Varsha. Thank you, Srishti Soni, for your nice presentation. Your topic was bioremediation of hydrocarbon polluted sites by using bacterial strain. She is pursuing MTech Biotechnology from ICRIWESA University, SATNA, and her research guide is Professor Aswini Pao, Madam. Now I would like to invite our next participant, Varsha Singh from BTech 8 Biotechnology. She is also pursuing degree from ICRIWESA University, Satna, and her guide is Dr. Mahi Chaure, ICRIWESA University, Satna. Now, Varsha,
Excuse me,
start
sir.
the presentation.
Excuse me. Session chair question round is. सर हो गया क्या क्वेश्चन राउंड सेशन चेयर का?
नहीं अभी नहीं हुआ.
अच्छा सर, प्रेजेंटेशन के बाद you will please ask the question to the participant and then next presentation will be there.
Yes.
Okay.
हाँ.
Miss Srishti, are you there?
Yes, sir.
Srishti, you presented very well your presentation. Bioremediation, enhancement of hydrocarbon contaminated sites by using an indigenous bacterial strains. Okay. So, what was the novel result of your presentation? Your research work? Can you explain?
So the noble result that I have noticed was the comparison between the free culture and the immobilized beads. The better result was shown by the presence of an immobilized beads, as the beads were better supportive for the growth of the bacteria and promoting their nutrition supportability. They were not affecting the bacterial directly. This oil was not directly in contact with the bacteria, and it supports the growth of bacteria in better situation. Whereas in the free culture, the bacteria was heavily affected as it was in contact with oil directly, and they were not even allowed to take the nutrition in a equal and proper amount. That was as the novel result I have noticed in comparison.
Okay. That is the good environmental biotechnology research, and with the guidance of your mentor, please try to make publish this research. Okay, that will be very beneficial for your dissertation and your career also. So
Yes.
try to publish this research in a good reputed journals. Scopas are a very indexed journals. Professor Wow is a very competent person. She will guide you. Okay.
So with
Okay, sure.
due permission of session chairperson, can I ask one question from Sasti?
You can ask. Okay.
Sasti, when you are doing this experiment or performing this research activity, could you
Yes,
tell
sir.
me which type of bacterial strain is most effective in degrading this hydrocarbon? Because I am also
Sir,
performing these activities on plastics. So have you identified any specific bacterial strains which are most powerful or most efficient for degrading this hydrocarbon?
Sir, Bacillus subtilis was being defined as the most strong bacteria. Comparison to the Pseudomonas aeruginosa, the one I have worked with, that was the bacterial name I have read in the
Have you
previous
isolated
research.
or done pure culture of that bacteria?
Which bacteria? Sir, Pseudomonas aeruginosa was the one I have worked with. So yeah, I have cultured those bacteria in the pure form.
Okay, Professor, you are call the second next participants.
Yeah, sir. She is already ready. I have announced her name 2 times. Now, Varsa, start your presentation and
Yes.
be punctual and limited in time, na?
5 minutes only. Varsa,
8
you have.
minutes. Yes, sir. 8 minutes and 2 minutes for theory session.
Okay,
8 minutes.
Okay, sir.
Beta, keep your phone in horizontal position.
Good morning, sir. My name is Varsha Singh. My thesis topic is assessment of antimicrobial properties of endophytic EMS1 against Fusarium oxyforum and human pathogens. This research focused on isolation and beneficial endophytic bacteria from plant tissue and evaluating their potential to inhibit plant pathogenic fungus and disease causing human pathogens. I want to introduce my little topic about my thesis. The Grand 9 banana is one of the most widely cultivated banana varieties in the world. The G9 banana is a highly popular because it's high yield, good fruit quality. Key point: scientific name of this banana Musa acuminata, common name Grand 9, and plant height 6 to 8 feet. Crop cycle 11 to 12 months from planting to harvest. Typical yield 25 to 30 kg per bunch. So let's know little about endophytic bacteria. Endophytic bacteria are microorganisms that live inside plant without causing any harmful disease to host plant. These bacteria are commonly found in root, stem, leaves, and seeds of many plants. Endophytic microbes produce various bioactive compounds such as bioactive enzymes and secondary metabolites. These compounds helps in protecting plants from pathogenic microorganisms and improving plant health. I explain about some fungus pathogens. The fungal pathogen Fusarium oxyvorum causes severe wilt disease in many crops and lead to significantly agricultural loss. human pathogens such as E. coli, Staphylococcus aureus, and Pseudomonas aeruginosa are responsible for various infection in humans. Isolation of EMS1. EMS1 is an endophytic bacterial strain isolated from plant tissue. It is capable to produce bioactive metabolites. These metabolites may inhibit fungal and bacterial pathogens. Morphological characterization. Morphological identification. Gram staining technique perform for morphological and physiological characterization of microorganism. It was observed under microscope. The purple violet color indicate gram positive and red pink color was indicating gram negative. This slide show result. Some are positive and rot safe bacillus. Biochemical test. Biochemical test on the basis of biochemical characterization. The bacterial isolates were biochemically characterized by some common biochemical test. Firstly, catalase test. It is used to detect of bacteria to produce enzymes that break hydrogen peroxide into water and oxygen, forming bubble formation. Indole test that determines the ability of bacteria to produce tryptophan to produce indole. Methyl red test detect mixed acid fermentation by bacteria producing stable acid from glucose. Next is bogus Prosker for identifying bacteria that produce acetone during glucose fermentation. Citrate utilization test determine whether bacteria can use citrate as their sole carbon source. I prepare a semen citrate agar. It was showing after autoclaving green color and pH is 6.7, sorry 6.9, and after bacteria utilize this citrate positive color pH will be 7.6 after and next this is my result. Some are positive and some are negatives. Next, plant growth promoting rhizobacteria PGPR. PGPR tests are done to check whether a bacteria can promote plant growth by improving nutrient availability, producing plant growth hormones. These tests are important in biofertilizer development, sustainable agriculture, and bio and soil microbiology. So I am firstly test included IAA production test. IAA full form is indole 3-acetic acid. It is used to detect the ability of bacteria to produce indole 3-acetic acid, a plant growth promoting hormones. Second, phosphate solubilization test. It is used to check the ability of bacteria to solubilize insoluble phosphate into solubilized form for plant uptake. Nitrogen fixation used to detect the availability and of bacteria to fix atmospheric nitrogen into capable from nitrogen like ammonia for plant growth. This picture is showing result of this PGPR test. Ammonia test to detect ability of bacteria to produce ammonia. Thank you, sir.
Ms. Varsa, you have presented a good presentation of your MTech, BTech thesis project work assessment of antimicrobial property of the endophytic EMS1 against Fusarium. So that question I want to ask you, EMS1 you have isolated from the plant tissue. Which plant tissue you have choose for the session of that? What,
Sir, banana plant.
Banana plant.
Yes, sir.
Okay. So in result you have you told there are a lot of some are the positive bacteria, some are the negative bacteria. So you have to say about that which bacteria are the positive and which bacteria are negative, and which are the very useful for the your growth of the different plants and that nutrition PGPR. Which bacteria are positive? Please tell me some name. Okay. EMS 1, 2, 17 and control. Control, you explain about the control. Which type you can choose which plant are what way about the control? Can you say?
सर कंट्रोल कंपेयर करने के लिए यूज करते हैं, कंटामिनेशन चेक करने के लिए.
नहीं, ठीक है. तो कहाँ क्या यूज किया था कंट्रोल के लिए? क्या साइड यूज की थी, सॉइल यूज की थी, प्लांट यूज की थी, क्या यूज किया था कंट्रोल के लिए?
सर प्लांट.
कौन सा प्लांट, कहाँ का प्लांट लिया था? चलो, दूसरा क्वेश्चन ये है कि यू टोल्ड कि पीएच इंक्रीज 6.9 to 7 या 6.5 to 7. तो कैन यू टेल व्हाट वाज द फैक्टर टू इंक्रीज द पीएच?
सर जब हम इसमें मतलब जो प्लांट बनाते हैं इसमें, तो जब हमारा जो बैक्टीरिया होता है तो जो मीडिया को यूटिलाइज कर लेता है तो उसका पीएच इंक्रीज हो जाता है.
मीडिया यूटिलाइज करने पीएच इंक्रीज हो जाता है. कितना इंक्रीज हुआ?
सर 7.6 हो गया था.
6.9 to 7.6 तो काफी ज्यादा इंक्रीज हो गया.
तो सर तभी तो उसका रिजल्ट पॉजिटिव दिखाता है. अगर नहीं होता है तो वो जितना रहता है उतना ही रहता है, ग्रीन ही दिखता है रिजल्ट, उसका कलर भी चेंज होता है.
अच्छा, और जो आपने हाउ मेनी पीजीपीआर टेस्ट यू हैव कंडक्टेड ड्यूरिंग द योर वर्क?
सर अभी मैंने कुछ किए हैं. आईए प्रोडक्शन टेस्ट किया है, फॉस्फेट, नाइट्रोजन फिक्सेशन, HCN एंड अमोनिया. सर अभी 2 टेस्ट बाकी हैं करने के लिए.
तो व्हाट वाज द रिजल्ट ऑफ द योर टेस्ट?
यस सर. सर उनके तो अभी रिजल्ट पेंडिंग है, इनके अभी मेरे पास हैं. सर ये पीजीपीआर टेस्ट के रिजल्ट है.
तो you did all the work in a lab, no?
Yes, sir.
Okay, good work, and you continue that works, no? And try to publish your work also.
Yes, sir.
Thank you, beta. Thank you, and Professor Paras.
Yes, sir.
Call the next participant.
Thank you, sir.
Thank you, Varsha. Now I would like to invite Sakshi Misra, BSc 8th Sem, BSc Honors 8th Sem, Biotechnology. Yes, sir. BSc 8th Sem, Biotechnology. BSc Biotechnology. Her topic is assessment of microbial properties of endophytic, isolation and characterization of plant growth promoting rhizobacteria from cement affected agriculture soil under heavy metal stress. She is also doing her research work with Dr. Mahi Chore, Assistant Professor, Biotechnology. Now over to you, Sakshi Misra. Start your presentation.
Sakshi, you have 5 minutes for your presentation and 2 minutes for discussion. Your time starts now. No, you have to conclude within the 5 minutes. Okay.
Good morning, present. Good morning, everyone.
Continue, beta. उसको बोले हैं कि सर ये अपना हॉरिजॉन्टल वर्जन कर ले.
My topic is isolation and characterization of plant growth, promoting rhizobacteria from cement-affected agriculture soil under heavy metal stress. I am comfortable in Hindi more, so I want to give my presentation in Hindi as well as in English.
Yeah. I'm comfortable in any language with
सीमेंट इंडस्ट्री के आसपास सीमेंट इंडस्ट्री के आसपास की जो एरिया एग्रीकल्चर सॉइल होती है उसमें सीमेंट डस्ट और हैवी मेटल्स जैसे लेड जिंक वगैरह जो मिक्स हो जाते हैं, इससे सॉइल की फर्टिलिटी कम हो जाती है और प्लांट्स की ग्रोथ पर नेगेटिव इफेक्ट पड़ता है जिसके कारण से क्रॉप्स पे भी बहुत सारे इफेक्ट पड़ते हैं. इसमें इस रिसर्च में जो सीमेंट अफेक्टेड एग्रीकल्चर सॉइल के जो रेजो स्पेयर एरिया प्लांट रूट के आसपास की जो सॉइल है उससे पीजीपीआर बैक्टीरिया आइसोलेट करके रिजोस्फेयर में नॉर्मल ऐसे बैक्टीरिया मिलते हैं जो प्लांट की ग्रोथ को नॉर्मली सपोर्ट करके ये बैक्टीरिया प्लांट को डिफरेंट तरीकों से हेल्प करते हैं. फर्स्टली मैंने सॉइल सैंपल कलेक्ट करके उसको आइसोलेट किया है और फिर आइसोलेट करने के बाद जो भी टेस्ट ये सॉइल सैंपल कलेक्ट करके आइसोलेट उसके मार्फोलॉजिकल कैरेक्टर पता करने के लिए मैंने आइसोलेशन की. आइसोलेशन के बाद स्टेनिंग करके जो भी बैक्टीरिया के शेप, कलर और साइज का पता चला. जो भी बैक्टीरिया प्रेजेंट थे वहां पर उसके स्टेनिंग करके हमने पता किया. हम बैक्टीरिया के कैरेक्टर पता करने के लिए करते हैं जो कि हमें बताता है कि कौन से बैक्टीरिया ग्राम पॉजिटिव है या नेगेटिव है. इसके बाद बायोकेमिकल टेस्ट लगाए हैं इसमें भी बैक्टीरिया के कैरेक्टर्स पता करने के लिए हमने बायोकेमिकल टेस्ट भी लगाए और बायोकेमिकल लगाए, फिर पीजीपीआर टेस्ट भी किए. ये ग्राम स्टेनिंग की मैंने और इसके जो बैक्टीरिया है दोनों आए पॉजिटिव और नेगेटिव. फिर एमआरपी जो टेस्ट किया हमने उसमें भी दोनों ही पॉजिटिव और नेगेटिव दोनों ही आए. ये पॉजिटिव टेस्ट आए। फिर कैटलेस टेस्ट भी किए, जिसमें कैटलेस में भी पता चला कि ये बैक्टीरिया जो है वो ग्राम पॉजिटिव बैक्टीरिया है, जिसमें बबल फॉर्मेशन हुआ था। इंडोल टेस्ट भी किए, जिसमें भी अभी जिसके रिजल्ट पेंडिंग है, उसका रिजल्ट अभी नहीं आया है। सिट्रेट टेस्ट भी किया, जिसमें कलर चेंज के अकॉर्डिंग उसका पता चलता है कौन से बैक्टीरिया है पॉजिटिव, नेगेटिव। और ये मैंने टेस्ट लगाए थे, जिसमें से पॉजिटिव थे और कुछ ने अपना इफेक्ट कम कुछ नेगेटिव, कुछ पॉजिटिव, और कुछ का अभी रिजल्ट नहीं आया है पूरी तरह से। मैंने पीजीपीआर जितने भी टेस्ट लगाए सर, उसमें बहुत सारे रिजल्ट अभी पेंडिंग में हैं। फिर पॉल्यूटेंट सर, फाइनल गोल हम लोग का ये है कि सीमेंट अफेक्टेड सॉइल से जो स्ट्रेस टॉलरेंस पीजीपीआर आइसोलेट करके उनकी प्लांट ग्रोथ प्रमोटिंग एबिलिटी को स्टडी करना है। थैंक यू।
ठीक, साक्षी, आपने अच्छा काम किया है।
यस सर।
इस पे कुछ चीजें
यस सर।
अभी आपकी क्लियर नहीं हो पा रही थी कि आपने कौन कौन से टेस्ट लगाए। आपने ये कहा कि बहुत सारे टेस्ट लगाए, तो बहुत सारों में क्या होता है, आपको जब वर्क करते हैं ना तो स्पेसिफिक चीज़ें बतानी पड़ती है कि हमने ये टेस्ट लगाया, ये टेस्ट लगाया, ठीक है, तो आपको वो बताना पड़ेगा। और बहुत सारे बैक्टीरिया प्रेजेंट थे, तो प्रेजेंट रहते हैं, तो आपने जो टेस्ट लगाया उसमें कौन से बैक्टीरिया प्रेजेंट थे आपको ये बताना है, ठीक।
सर, मैंने अभी जितने भी टेस्ट लगाए सर, उसमें ग्राम पॉजिटिव और नेगेटिव दोनों ही प्रेजेंट थे।
कौन से बैक्टीरिया ग्राम पॉजिटिव थे, कौन से बैक्टीरिया ग्राम नेगेटिव थे, ये आपको यहाँ पे बताना चाहिए, ये बोल रहा हूँ मैं, है ना। आप ये नहीं बोलिए जितने भी टेस्ट लगाए, आपको ये बताना पड़ेगा कि हमने कितने टेस्ट लगाए, 4 लगाए, 5 लगाए, 10 लगाए, तो यू हैव टू टेल द नंबर ऑफ द टेस्ट, जितने भी नहीं, क्योंकि आपने रिसर्च कर रहे हैं ना, तो हमें एक संख्या देनी पड़ेगी, ठीक है।
ओके सर।
हाँ बोलो।
सर, अभी उसका रिजल्ट नहीं आया है ना, रिजल्ट नहीं आया है, इस वजह से अभी सर आइडेंटिफाई नहीं कर पाए हैं।
अच्छा, तो दूसरा भी आपने एक बड़ा बोलने में गड़बड़ी होगी, बबल्स आया तो ग्राम पॉजिटिव है, तो बबल्स आना ग्राम पॉजिटिव होने का क्या ये है क्या? ये कौन सा टेस्ट है? यदि बबल्स आ जाएंगे तो ग्राम पॉजिटिव हो जाएगा, बबल्स नहीं आएंगे तो ग्राम नेगेटिव बैक्टीरिया हो जाएगा।
कैटलेस टेस्ट है, इसमें ग्राम अगर बबल्स प्रेजेंट है उसमें तो वो पॉजिटिव है, अगर प्रेजेंट नहीं हो रहा है तो वो बैक्टीरिया नेगेटिव है।
अच्छा, तो इसको आपने देखा है तो कौन कौन से बैक्टीरिया हैं इसमें? अभी रिजल्ट नहीं आया, अभी है ना?
सर, अभी रिजल्ट नहीं आया है।
ठीक है, कितने पीजीपीआर टेस्ट लगाए आपने?
सर,
संख्या
अभी सिर्फ दो लगाए हैं। फॉस्फेट सॉल्युबल टेस्ट, आईए टेस्ट, इंडोल 3 एस्टिक एसिड और अमोनिया टेस्ट।
तो 4 टेस्ट लगाए, तो अभी इनके रिजल्ट्स क्या शो कर रहे हैं?
सर, अभी इनके रिजल्ट नहीं आए हैं।
चलो गुड, अभी तो आपका काफी समय है, तो इसको आप अच्छे से इसको रिजल्ट लीजिए, अच्छे से कंक्लूजन करिए, ठीक है ना?
यस
ओके, थैंक यू, गुड प्रेजेंटेशन। नेक्स्ट।
Thank you, Sakshi, for your nice presentation. Next, I would like to invite Sanjana Soni, BTech 8th SEM Biotechnology from AKS University, SATNA. She is doing her research work with Dr. Kamlesh Soni. Her topic is comparative in silico and transgenic investigation of tobacco, DNA JV, and Arabidopsis 80 DJB3 reveals a conserved stress chaperone network. Now, Sanjana, let's start your presentation.
Good morning to everyone. Sir, स्लाइड दिख रही है?
हाँ दिख रही है।
इसको थोड़ा बड़ा कर लो, उसको F5 दबा के उसको एनलार्ज कर लो।
Yes, sir.
Yeah, nice. Now start your presentation.
Okay. Sir, my topic is. My topic is comparative in silico and transgenic investigation of tobacco, DNA GV, and Arabidopsis reveals a conserved stress chaperone network. So introduction is climate changes due to global warming causes various abiotic stresses. These causes the abnormal temperature, soil, drought stresses to the plants. Heat, a major abiotic stress of plants by global warming, incurs irreversible damages which hampering plant photosynthesis, development, and reproduction. Why heat shock proteins are used? So heat shock proteins play a key role in controlling the various stresses. HSPs are classified as their approximate molecular weights, such as HSP 100, 90, 70, and so on. HSPs are mainly why HSP? HSP also known as DNA J proteins and constitute DNA J HSP40 family and are conserved as a co-chaperone for HSP70s. The diagram shows how HSP works and works in refolding of unfolded proteins. Research gap. No in silico analysis इसकी कोई भी सिलिको एनालिसिस अभी नहीं हुई है और कोई इसका प्रमोटर एनालिसिस नहीं हुई है. इसका जो एम है, द एम इज इन सिलिको एनालिसिस ऑफ निकोसियाना टोबैकम HSP40 एंड रोल ऑफ Arabidopsis thaliana HSP40 in tobacco. By HSP40, so it controls the stress in plants. The diagram shows how HSPs are helps in refolding of proteins. इसमें ये बता रहा है कि जब हीट स्ट्रेस होता है प्लांट्स में तो जो प्रोटीन उसके अनफोल्ड हुए हैं तो वो उससे एक सिग्नल मिलता है HSPs को, तो HSPs के जीन्स को, तो फिर देन HSPs के प्रोटीन्स जो हैं वो जाकर अनफोल्डेड प्रोटीन से बाइंड होते हैं और उसकी रिफोल्डिंग करते हैं. प्रोटीन सीक्वेंसेस, स्लाइड में प्रोटीन सीक्वेंसेस की एनालिसिस की गई है जिसमें ये प्रोटीन जिसमें ग्लाइसिन का जो हाई परसेंटेज है रहा है और जो सिस्टीन है उसका सबसे लीस्ट परसेंटेज रहा है. और इसके लिए हमने एक्सपेसी प्रॉट परम टूल का यूज किया है. फाइलोजेनेटिक एनालिसिस ऑफ निकोसियाना DNA J, तो इसमें जो है मेगाटॉल सॉफ्टवेयर का यूज किया गया था ट्री को यूज करने के लिए और इटॉल टूल का यूज किया था इस ट्री को मॉडिफाई करने के लिए. और जो हीट मैप है उसको बनाने के लिए हमने गूगल कोलैब का यूज किया था. और इस पूरी एनालिसिस में ये फाइंड हुआ है कि जो हमारा निकोसियाना DNA J है उसकी सबसे ज्यादा हाईएस्ट सिमिलैरिटी कैप्सिकम एनम में पाई गई है. प्रोटीन इंटरेक्शन एनालिसिस ऑफ एंटी डीएनए जेम। तो इस एनालिसिस के लिए हमने स्ट्रिंग डेटाबेस का यूज किया था, और ये जो टेबल है इनमें ये जो डायग्राम है वो ये शो कर रहा है कि जो भी हमारा डीएनए जे प्रोटीन जो है HSP40 वो कौन कौन से प्रोटीन के साथ इंटरेक्ट कर रहा है। तो ये इसकी आईडी, लोकस आईडी दी हुई है और उन प्रोटीन्स के नेम दिए हुए हैं। डीएनए जे प्रोटीन का ये बायोलॉजिकल रोल है और मॉलिक्यूलर फंक्शन इसमें शो हो रहा है, जिसमें बायोलॉजिकल फंक्शन में जो ये सर्कल है वो बता रहा है नंबर ऑफ प्रोटीन्स को, और जो कलर है वो लाइट कलर जो शो कर रहा है मोस्ट सिग्निफिकेंट रोल को शो कर रहा है, जिसका जो कि बायोलॉजिकल फंक्शन में इसका प्रोटीन फोल्डिंग में और सेलुलर रिस्पॉन्स ऑफ हाइपॉक्सी और रिस्पॉन्स टू एबायोटिक स्टिमुलाई में इसका जो है रोल देखा गया है। मॉलिक्यूलर फंक्शन जो है इसका वो एटीपी डिपेंडेंट प्रोटीन फोल्डिंग में एटीपी बाइंडिंग और चैपराउंड बाइंडिंग में देखा गया है। ये इसका सबसेलुलर लोकलाइजेशन बता रहा है। इस एनालिसिस के लिए हमने वॉल्फ पीसॉर टूल का यूज किया था। प्रमोटर एनालिसिस, जो ये डीएनए जे प्रोटीन की जो भी प्रमोटर है इसके हमने 1.5 kb सीक्वेंसेस को एनालाइज किया है, और ये सीक्वेंस है इनका क्या फंक्शन है वो इस पे बता रहा है। ये जो है ये सिस एलिमेंट्स को बता रहे हैं। डेवलपमेंट ऑफ ट्रांसजेनिक इन टोबैको, ये जो डायग्राम शो कर रहा है इसमें ये जो भी हमारा कंस्ट्रक्ट है ये जीन कैसेट को बता रहा है, जिसमें पीनॉस और एनडीटीएल, पीनॉस कैनामाइसिन रेजिस्टेंस का प्रमोटर है, और जो एराब्डोप्सिस थालियाना डीएनए J3 जीन है P35S प्रमोटर इस पे यूज हुआ है। ये कैसेट हमने ये वेक्टर में इसमें टोबैको में हमने इसको इंट्रोड्यूस कराया है, और ये हमारा एक्सप्लांट प्रिपरेशन को कर रहा है। हम इसमें हमने एराबिडोप्सिस के डीएनए जे प्रोटीन को लिया है और उसको हम टोबैको के प्लांट में इंट्रोड्यूस करके उसकी एनालिसिस कर रहे हैं हेट्रोजीनस सिस्टम को स्टडी करने के लिए। कैलस फॉर्मेशन, फिर जो हमने एक्सप्लांट डेवलप किया था उसी में से लीफ डिस्क ट्रांसफॉर्मेशन मेथड का यूज करके हमने ट्रांसजेनिक अपने डेवलप कर रहे हैं, जो कि अभी कैलस स्टेज तक पहुंचा है और इसकी अभी फर्दर एनालिसिस होगी। Conclusion, anti-DNAJ saw many regulatory elements in the promoter region, and anti-DNAJ was predicted to be involved in stress responses. Way forward, once transgenics in tobacco is developed, the physiological and molecular function of AT-DNAJ will be characterized in a heterologous system. Okay, thank you.
So Sanjana, you presented nice work of your work research 8th semester. You are a student of 8th semester, no?
सर आपकी वॉइस नहीं आ रही है, कट रही है।
आप 8th semester के स्टूडेंट हो ना?
Yes sir, yes sir.
हाँ, one question कि why you choose the tobacco plant, not another plant?
सर इसको हम इजी लेबोरेटरी कंडीशन में मेंटेन कर सकते हैं।
आपने किया है?
और इट इज आल्सो अ मॉडल प्लांट।
तो आपने किया है इसको?
यस सर, कर रहे हैं हम।
कर रहे हैं कि किया है?
यस सर, किया है।
अच्छा, और क्या ये दूसरे प्लांट पे भी हो सकता है? हम टोबैको ना ले सकते, कोई भी वेजिटेबल प्लांट्स लें, कुछ ऐसा लें तो?
यस सर, और टमाटर पे हमें इसको करना भी है अभी। एक ये टोबैको पे, फिर हमें इसकी एनालिसिस टोमेटो पे करनी है।
अच्छा, गुड, वही पूछना था। ठीक है,
Okay, sir. Thank you, sir.
ओके। Para sir,
Yes, sir.
call the next participant.
Yes, yes. Thank you, Sanjana. Now I would like to call Neha Patel, 2M.Tech Biotech from AKS University, SATNA. She is also doing her research work under the supervision and guidance of Dr. Kamlesh Kumar Soni. Her topic is putrescine-mediated stress-induced changes in mung bean. Over to you, Neha Patel.
Good afternoon, everyone.
Neha, you you complete your presentation within the 5 minutes, okay?
Okay, sir.
हाँ.
My topic is amelioration of salinity stress by exogenous application of putrescine in mung bean. A plant faces different stress during the changing environment. A high level of salt result ionic imbalance, osmotic stress in plant, which causes severe effect on morphology, biomass, and biochemical processes of the plants, and ultimately result in plant damages. Salinity stress is one of the most notorious environmental factors restricting the productivity of mung bean in arid and semi-arid regions. Why mung beans? Mung beans are important food grain legume crops. Mung bean is an essential legume that has been consumed as a functional food in many countries like China due to its high protein content. Putrescine, a polyamine, is an organic compound having a more than 2 amino acids. It is a low molecular weight aliphatic polyamine and it has 2 amino groups. Putrescine, here is a pathway, metabolic pathway of putrescine. Putrescine, it's regulate the TCA cycle of an organism, and it's affect the number of events in plants. Growth changes. It enhance the leaves number, shoot height, root length, root elongation, and also play an important role in biochemical changes like increasing our hormones labeled as IA, CK, cytokinins, and carotenoids, vitamin C, and soluble sugars. Objective: can putrescine overcome the stress of mung bean plants? Strategies: seed viability test. We are performing seed viability test treatment of seeds and plants with different NaCl concentration to see the maximum effect. And we are performing a phenotypic and physiological changes. Seed viability test technique used to determine whether individual seeds appear to be dead and alive within a sample. Number of seeds germinates and total number of seeds sown. कितने number of seeds हमारे हैं और कितने number of seeds germinates हुए हैं. मतलब for example 120 seeds and seed variety is 92%. Effect of salt stress on seed germination. We have seen in this picture control the germination to a high level and 25 millimolar concentration. We in 50 and 100 millimolar concentration, the salt stress is high. Observable effect of salt stress in germination. Germination is also affected by this salt stress. Significant effect was observed at 50 millimolar and 100 millimolar NaCl concentration. Continue with 50 millimolar and 100 millimolar NaCl. Seed effect of putrescine on seed germination. In this picture, the control and 50 molar Putrescine is also reduce the stress in 50 millimolar and 100 millimolar. And here is a graph representation. And water supply of leaf and transpiration rate. Putrescine also play in role in water. And effect of putrescine on shoot and root length. In shoot and root length, putrescine reduce the salt stress and enhance shoot length and root length of the mung bean and plants. Conclusion, significantly improvement has been observed on growth and development of plant by exogenous application of putrescine. The study provides a foundation for further analysis of stresses plant on application of putrescine. Detailed analysis by looking at biological level and molecular level will verify the observation made in the presented study. Thank you.
Neha. Okay.
Yes, sir.
Neha, you did a good work. And
Thank you, sir.
one question is that
Yes, sir.
control site and the different concentration of the NaCl. No? I have seen your presentation and one slide. Control site has the maximum growth, no? And the NaCl concentration media was the minimum growth.
Yes,
Right?
sir.
And how you can reduce increase the root length and root weight length? Explain.
सर आपकी आवाज थोड़ी क्रैक हो रही है.
ये बोल रहा था बेटा, कंट्रोल आपने जो प्रेजेंटेशन दिया है एक स्लाइड में दिख रहा था कि कंट्रोल साइट पे आपका अच्छा ग्रोथ हो रहा है, ठीक है ना?
यस सर.
और जो आप NaCl कंसंट्रेशन लिए उसमें से ग्रोथ कम है, तो उसका एक स्पेसिफिक क्या कारण क्या हो सकते हैं, बता सकते हो?
क्योंकि जो NaCl है वो जो उसका जो वाटर है उसको इवोपरेट मतलब एक्सोजेनसली बाहर कर देता है.
ओनली वन रीजन कि और कुछ भी है? आपने इसको पढ़ा है, रिव्यू पढ़ा है, कुछ पेपर पढ़े हैं?
यस सर, अभी तो मैंने एक ही रीजन को लेके ये सारी चीजें की हैं.
अच्छा, और किस कंसंट्रेशन में आपका रूट का और शूट का जो लेंथ है वो ज्यादा हुआ है? NaCl के किस कंसंट्रेशन?
100 मिलीमोलर.
100
मिलीमोलर जो कंसंट्रेशन थी उसमें ज्यादा शूट लेंथ एंड रूट लेंथ देखने को मिली.
और कंट्रोल पे
तो वो तो होगा ही ना, क्योंकि उसमें कोई स्ट्रेस ही नहीं है और वो भी नहीं है, तो इसलिए उसकी सबसे ज्यादा होगी.
अच्छा, तो 100 पे आपने किया और नीचे 50 पे किया और कितने पे किया है?
25 मिलीमोलर, 50 मिलीमोलर, 100 मिलीमोलर.
तो आपका जो पॉलीएमीन्स है तो इसमें क्या इसमें जो करने पे क्या आपको रिजल्ट की फाइंडिंग क्या मिली और आगे क्या फर्दर वर्क आपने बताया करना है, तो क्या मेजर फाइंडिंग क्या मिली आपको?
मेजर फाइंडिंग सर मुझे अभी इसमें ये मिला है कि ये जो पॉलीअमाइन पुट्रेसीन होता है इसका रोल ये स्ट्रेस को रिड्यूस करने में ये अच्छा रोल प्ले करता है, गुड रोल प्ले करता है.
ठीक है सर. नेक्स्ट कॉल टू नेक्स्ट पार्टिसिपेंट.
Thank you, Neha. Now, I would like to call Yogeshwari Pandam, MTech Biotechnology 4th SEM. She is doing her internship with Dr. Kamlesh Soni on topic in silico characterization and preliminary transgenic analysis of AT-DJB3A DNA J chaperones involved in stress tolerance in tobacco. Over to Yogeshwari. Please conclude topic quickly within 5 to 6 minutes. Next participant, Priyanka and Varsa will be ready, no? For their poster presentation. Yogeshwari,
Yes, sir.
are you ready, beta?
Yes, sir.
Then start your presentation.
Yes, sir.
Beta, please start your presentation.
Sir. Hello. Hello.
Beta, continue. Your voice is coming and आपकी स्लाइड नहीं शो रही बेटा, स्लाइड शो करिए.
बेटा उसको इनेबल एडिटिंग में जाके जब जाओगे उसके बाद डिजाइन में जाके फुल स्क्रीन मोड पे करो उसको, तब स्टार्ट करो. योगेश्वरी, यहाँ इनेबल एडिटिंग पे जाओ पहले. स्टार्ट करो. Hello, Priyanka, are you ready for your poster presentation?
Yes, sir.
Then start your. We'll continue with Yogeshwari in the next, after both of you. Start your poster presentation.
Sir, is it my screen is visible to you all?
Yeah, but little bit elaborate it, no?
Yes,
It's
sir.
so minute we can't able
Yes,
to
sir.
see the letters.
Yes, sir. Yes, sir. Okay. So now it is fine.
बेटा आप इसको जो है ना उसका परसेंटेज बढ़ा दीजिए.
Okay, sir.
The topic of Priyanka Parmar is effect of growing season on growth, flowering, and yield attributes of sponge gourd.
So shall I start?
Yeah, first introduce yourself because we don't know from where you are and all that. Please introduce yourself, then start your presentation. No?
Okay. Good afternoon, all. Today, I, Priyanka Pawar from Govind Ballabhan University of Agriculture and Technology, is going to present my poster seminar on effect of growing season on growth, flowering, and yield attributes of sponge gourd genotype. First of all, a brief introduction is that sponge gourd, it is known as Torai, Gilgi Torai, and Nenua in Hindi, and it is an important cucurbitaceous vegetable widely cultivated in tropical and subtropical areas of India.
Sorry,
And
beta,
it
but
belongs.
your screen is not visible. Please share your screen.
Okay. Sir,
Share your screen.
Okay, sir.
Yes, beta, now it is visible. But increase your size a little bit more if possible.
Okay, sir. So now is it visible?
साइड वाला उसको F5 करो बेटा तो आ जाएगा. F5 करो.
हाँ.
So is it visible?
No बेटा बंद हो गया.
Okay, so now.
You have stopped your presentation. Okay, start. Scroll down and scroll up and start.
So is it visible now?
Okay, start. Yes, visible. Please start.
Okay, good afternoon, everyone. Today, I, Priyanka Pawar, is going to present my poster seminar on effect of growing season on growth, flowering, and yield attributes of sponge gourd genotypes. Now, the introduction. Sponge gourd is also known as Torai, Gilki Torai, and Nenua in Hindi, and it is an important cucurbitaceous vegetable, mostly widely cultivated in tropical and subtropical region. It belongs to the family of Cucurbitaceae and grow for its tender fruits, and which is very highly nutritious and highly rich in carbohydrate, vitamin C, minerals like magnesium, calcium, and zinc and iron, and also exhibit the notable pharmaceutical properties like antioxidant, anti-cancer, and anti-inflammatory activities, which making it highly beneficial for the human health. And it is also used as a immature fruit and mature fruit also. Now the material method of my poster is that the experiment I was conducted during the Zaid and Kharif season of 2025 at VRC of Department of Vegetable Science, Govind Ballabh University of Agriculture Technology, and the trial was laid out in a randomized complete block design, in which the 12 sponge gourd inbred lines were selected for the evaluation of an observation. Now, there are the result and discussion of my seminar is that table number 1 mainly present the mean performance of a sponge gourd genotype for the flowering parameter. In this table, first variety, the Kashi Shreya, mainly taken the minimum time for days to first female flower appearance, male flower appearance, and days for the first harvesting. As compared to the Zaid season, and the fourth one is the days to last harvesting. Pusa Sneha shows the longer blooming period of time for the flowering and for increasing the yield productivity of a crop. And the fifth one is the sex ratio. Pusa Sneha shows the minimum sex ratio as compared to the all genotypes in Zaid season. Now the table number 2, it shows the growth and yield parameter performance of a sponge gourd, in which Swarna Prabha genotype shows the maximum vine length in case of Zaid season, while Pusa Sneha shows the maximum vine length in Kharif season. In case of number of primary branches, Kashi Shreya shows maximum number of primary branches in Zaid season, while Kashi Shreya shows maximum in Kharif season. In case of fruit length, Pusa Chini shows maximum in Zaid season, while Pusa Sneha shows maximum in Kharif season. In case of average fruit weight parameter, Kashi Shreya shows maximum in Zaid as well as in Kharif season. And in case of fruit diameter, Pusa Chikni shows the maximum in case of Zaid as well as in Kharif season. In last fruit yield per plant, Pusa Sneha shows maximum in Kharif season, while Kashi Divya shows maximum in Zaid season. Now the conclusion of my study is that it revealed the growing season significantly influenced the fruit yield as well as the flowering and growth parameters of a sponge gourd genotype, and the overall higher fruit yield per plant was observed during Kharif season compared to the Zaid season. And the identified superior genotype can be recommended for season-specific cultivation and also future breeding programs. Thank you so much.
Miss Priyanka,
Yes, sir.
you told you have selected the 12 sponge gourd,
Yes, sir.
and how many varieties have you selected? Genotype?
Sir, 12 genotypes are selected.
Uh, and which, as per your result, which genotype
Yes,
is
sir.
the more efficient for their food's weight and food's length, nutritional values? What is your the
Uh,
result?
Sir, mostly the Kashi Shreya shows the better performance in flowering parameter, like it create a earliness in the market. And in case of growth parameter, mainly Pusa Chikni and Kashi Shreya and Kashi Pusa Sneha shows maximum growth for Zaid as well as Kharif season. While in case of yield, we prefer mostly which can give the earniness to the market and more profitable to the farmer as well as for the person. I recommend Pusa Sneha as well as Kashi Divya variety.
Okay, so because the farmer don't know their botanical name and their characters, so as a
Yes, sir.
researcher, you will suggest that only two genotypes variety to the farmers, so they can gain the more benefits, more income from these crops. Okay.
Yes, sir. Yes, sir.
Okay, and so what was the difference in their fruits, in their color? One thing we have also noticed that some fruits are in the taste, bitter taste. Why?
Yes, sir.
What was the reason behind that?
Sir, because Turai mainly contain the solosodium compound. Because we know that मतलब जब हम उसको कट करते हैं तो हम देखते हैं कि उसमें ब्राउनिंग स्टार्ट होने लग जाता है, उसके बाद वो मतलब बिटरनेस टेस्ट क्रिएट करता है.
Okay, so you performed your research in the Govind Ballabhbhai Pant University.
Yes, sir.
Okay, good. Good.
Thank you so much, sir.
Sir, please call the next participant.
Yes, sir, yes. Now I would like to call Varsha Ratan for her poster presentation on harnessing liquid organic manure for soil health and crop productivity. Varsha,
Yes, sir.
are you ready?
Yes, sir. So just wait. I'm sharing my slide.
First, introduce yourself because there is no your profile in our
Okay.
record. So, from where you belong to, from where you are pursuing your degree. Just brief.
A very good afternoon to one and all present here. I have done my doctorate from CSK HPKV, Palampur, Himachal Pradesh, and at present I am working as assistant professor in DAV University in Agriculture Sciences Department, Jalandhar.
Yeah.
So, is my slide visible?
Yeah, visible, but enlarge it a little bit more. No?
So, is it visible?
Is it visible? But open your this slide. So mail is visible actually. Open that and enlarge that.
So now is it visible?
1 minute, wait. It's presenting now. Wait. No, not visible till now.
So is it visible now?
Not
No.
visible.
So please wait. I'll just try again.
Your mail is visible, attachment is visible, but individually it's not visible.
First download that attachment, na beta?
Sir, I had downloaded it earlier, but just wait. I'll just see again.
Open from the downloads. Yogeshwari, are you ready with your presentation?
Yes, sir.
Wait for 1 or 2 minutes. If Pasha is able to open her poster, then she will continue. Otherwise, you will start.
So is my screen visible now?
Yeah, your screen is visible, but no. Open your document, open your poster from the downloads.
Just wait, sir. I'll just open. So is it visible?
Yeah, it is visible.
I'll just present. So now is it visible?
Yeah.
Zoom it. Please zoom it.
So shall I proceed?
Sir is saying that please zoom it. Because
Okay, zoom it. Okay,
Yeah,
sir.
yeah.
Is it fine now?
Yeah. When you are explaining, please zoom with the topic.
Okay.
So
Okay, so no
we can
problem.
able to see the small letters.
Okay, sir. Okay.
Yeah.
So I again greet all of you. A very good afternoon to all the respected judges and my fellow participants. My poster presentation is titled Harnessing Liquid Organic Manures for Soil Health and Crop Productivity. This is a review highlight of how liquid organic manures influence soil properties and improve crop performance across different horticultural crops. So the introduction part. Nowadays, modern agriculture is facing the challenges of maintaining soil health while sustaining crop productivity. So, with the growing need of sustainable agriculture, farmers are increasingly adopting liquid organic manures as eco-friendly alternatives to synthetic fertilizers. Excessive use of chemical fertilizers has increased yields in short term, but has also caused soil degradation, nutrient imbalance, and decline in microbial activities. Therefore, there is a growing interest in eco-friendly nutrient sources, especially liquid organic manures such as vermiwash, compost tea, etc. These contain readily available nutrients and beneficial microorganisms, enzymes, and various plant growth promoting substances that support both the soil as well as the plant health. So the objective of this study was of this review was to evaluate the effects of liquid organic manure on soil health and crop productivity based on findings from various studies conducted in different fruit and vegetable horticultural crops. So now I'll talk about the results of the various people, those who have, or various researchers, those who have worked on it. So first one was done in citrus fruits or citrus trees, which concluded the result showed that vermiwash significantly increased the uptake of macro as well as micronutrients. In maize, authors or the researchers like Moridi et al. In 2021, he resulted that the studies supported or reported that improvement in soil biological activities, soil porosity and aggregate stability, all of these soil physical parameters increased by using these liquid organic manures, and it also helped to reduce the bulk density of the soil. In onion, it was reported that the compost tea enhanced the plant growth, yield, and nutrient content of the crop. And in chilies, The researcher Ujjwal concluded that vermi-wash improved the various plant attributes like plant height, number of leaves, branches, and fruit characteristics, while reducing the days to flowering and harvesting. In soybean, it was observed that the yield and quality of the soybean was significantly increased by the foliar application of vermi-wash compost tea. And next is black gram, where the researcher Kumar et al. In 2021, they reported that application of vermi-wash from vermi-wash and significantly influenced the growth parameters and yield attributes, thus achieved maximum yield, this seed yield, all yield, and BC ratio. And Kevin did research work on tomato, and he concluded that he reported that chicken manure tea showed the highest growth and yield, followed by vermicompost tea and bioslurry, outperforming inorganic fertilizers while also offering better economic returns, indicating that organic manures can sustain profitable tomato production. So the conclusion, so based on this comprehensive review of the available data, liquid organic manures such as vermi-wash, compost tea have shown significantly or have significantly enhanced the crop growth, yield, and quality compared to inorganic fertilizers. They improve key growth parameters, accelerate flowering, and increase nutrient uptake while enriching soil fertility. These liquid manures not only boost productivity but also provide high economic returns, making them sustainable and a profitable option. Overall, they support eco-friendly farming practices and contribute to long-term agricultural sustainability. Thank you, sir.
Miss Versa.
Thank you, sir.
Dr. Versa, this is a good review presentation.
Yes, sir. Thank you, sir.
I want to know, just want to know from you,
Yes, sir.
a liquid organic manure is more effective than the solid that you want to say?
Yes, sir. No, sir, but it is equally, but it can be a better option as compared to the chemical fertilizers that we are using.
So organic manure may be in the solid form also.
Yes, sir. So organic, yes, sir. But sir, it is better to use in liquid form as compared to the solid form.
But the maintenance and the transportation and another thing is the more costly, and it is too adjustable.
Sir, yes, sir. Sir, I totally agree with you. But when we compare the solid forms, they are more bulky. So it is more difficult for us to carry all those. And in comparison to that, these liquid organic manures are less bulky. They are easily transportable, and the nutrient content and the absorption power of these nutrients is better as compared to the solid ones. Because sir, these entirely we can use them in the form of as a in fertigation process also. So ultimately they will go into the roots, and we can also use for the foliar application of these organic manures. So according to me, I think liquid organic manures are better option.
Okay, how many research papers you reviewed to write that review paper?
Sir, I have reviewed across 10 to 12, 20 papers. Even my PhD work, I had one component of vermi-wash and compost tea, which I am still writing the paper.
Okay.
Yes, sir.
And how many parameters you observed when you use the liquid organic manure, and which plants?
So my work was on tomato and cucumber. I did it under protected conditions, and actually I had two more factors. So along with that, I had these factors in which I did soil physical parameters were the main, because they were mostly affected by using of these liquid organic manures, as well as the nutrient content of the plant also was better.
So have you observed the growth parameters, fruiting or flowering is the more affected, the performance
So,
is better than
my focus entirely was on the soil physical properties.
Okay.
Yes, sir.
Okay, good.
Okay, thank you, sir.
But sir, I want to know.
One candidate, Yogeshwari Srikant Pandram, is able to present?
Yes, sir. Yes, sir.
Yogeshwari, you continue your presentation?
Yes, sir.
But sir, stop sharing your screen, na?
Okay, sir. Okay. Thank you, sir.
Yogeshwari, start your presentation.
Visible?
Yeah, it is visible.
Please press F5. Full presentation.
Is analysis of 80 AB3 carbon in states tolerant tobacco. Primary role in response technology his one of the most associated with global warming and causes disease and affect development and
So your voice is not clear.
The network problem is there.
Try once more.
I think she has technical glitch about the network. Please call next to present the PowerPoint. Next remaining.
Yes.
Paras sir,
Yes, ma'am. Yes, ma'am.
please call to the next participant.
I think participation may be completed.
All completed?
Yes.
No one is remaining?
No.
Okay.
Yes, ma'am.
I will call her, sir. I will call Yogeshwari to join.
Yes, ma'am. We will wait for just 1-2 minutes.
Yes, sir. Please wait.
Yes.
Sir, Yogeshwari is joined. Yogeshwari, are you there?
Yes, ma'am.
Please present your screen. Paras sir, Shreya Vishwakarma also completed her presentation.
Shreya Vishwakarma. No, there is no name of Shreya Vishwakarma.
She is in your list. Paras sir?
No, she is not in our list.
Okay. ठीक है sir, thank you.
Videshri? Yogeshwari?
Yes, sir.
Are you there? Start presenting,
Yes, sir.
Mita. This is the third time we are giving you opportunity.
Sir, actually, Kamlesh Soni sir is joining the meeting to present the slide of Yogeshwari.
Okay.
Okay. Yogeshwari, are you presenting? Okay. Let's continue.
Yes, sir.
Okay, continue.
Open your presentation on screen.
It a first close previous one, na? Multiple screens, so it is unable to sharing.
I think Professor Paras, I think
Yes.
it's a technical issue. Please.
Yes, sir. Can I conclude the session, sir? With your due permission?
You can conclude.
Thank you, sir. It's a great privilege to honor as a session coordinator in this technical session of the track 2 in International Conference on Research, Innovation and Women Empowerment in Sustainable Agriculture Development, ICRIWESA 2026, organized by Edu-Mentors Society for Sustainable Agriculture and Rural Development and Departmental of Biotechnology, FLST, AKS University, SATNA, as a great knowledge partner. First of all, I thank to the representatives and authorities which joined in our track 2 session, Mr. Astos and Arvinder Singh Pau sir, and our session chair, Dr. Mahendra Tiwari sir.
Excuse me,
Yes.
sir. Paras
Yes, ma'am.
sir.
Yes, ma'am.
कमलेश सोनी सर ज्वाइन हो गए तो करा लेते ना अपन उनका प्रेजेंटेशन, अगर सेशन चेयर की परमिशन हो तो.
Yes, sir.
महेंद्र सर, कैन
मैंने उन्होंने म्यूट किया लगता.
Paras sir?
Yes, ma'am.
Kamlesh Soni sir is presenting the screen and Yogeshwari will deliver the PowerPoint.
Yes, sir.
है ना? Sir, please present. Yogeshwari, you can unmute yourself and start the presentation. Quick.
Yes, ma'am.
Yogeshwari, whenever you need, just let me know. I will do the next slide. Okay. So this is your next slide. You start your introduction. Introduce yourself and then start your introduction.
Okay, sir. My name is Yogeshwari Shrikant. My topic is in silica characterization and preliminary transgenic analysis of ATJB3 and DNAJ capillodon involves in stress tolerance in tobacco. Introduction. These proteins are primarily involved in heat stress tolerance. They function as molecular capillodons to maintain proteins and folded states. Then change lead abnormal conditions such as temperature and stress. Third is heat stress, one of the major abiotic stress associated with global warming, causing irreversible damage to plant by affecting photosynthesis, growth, development, and reproductive process. So next slide. Why HSPs? HSPs is a heat shock treatment. Heat shock protein play a role protecting plant against various environmental stress. And their basic HSPs are classified in basic their molecular weight families include HSP100, HSP90, HSP70, HSP60, and small HSP. What is aim? In silico and functional study of AT-DNAJ in tobacco. And next slide. Then protein sequence of AT-DNAJ. The protein was found to be positive charge. This was highest amino acid, 11%. So next slide. Phytogenetic analysis. There are the two diagram. First is tree diagram, and the second is heat map. Mega 12 software was used to create the tree. iTool tools was used to modify the tree, and global CoBlab was used to make the heat map. And we know the highest similarity to the Pinsica, and that is the pomegranate. This is like the protein interaction analysis. There are the 5 IDs and interaction proteins are here. Then we know the protein dominance and features. Protein dominance feature, we show the diagram in the diagram between specific proteins, and there are the list of the dominance and structure in the protein help to identify functional motif that mediate interaction or biological roles. And then subcellular localization. They are the chart are located inside the cells, and localizing data guide understand of protein functions and interaction context. Then biological and molecular, then promoter analysis. These are the list of elements, a gene promoter that control expressions. Development of transgenic in tobacco. There are these are the figure diagram. There are the left border and right border, and these TNOS, this promoter derives the expression of downstream gene in plant, and then NPT2, and we use the leaf disc transformation method in tobacco, tangerine. Conclusion, AT-DNAG regulatory elements that promote the region. AT-DNAG was predicted to involve in stress response. For future work, once tangerine in tobacco is developed to physiological and molecular function of AT-DNAG will characterize in a heterologous system. Thank you.
So Yogeshwari, you presented your work.
Yes, sir.
I want
Yes,
to
sir.
know from your side, that is just one question, what you find, you showed the phylogenetic analysis and the heat map diagram. So please explain me, what is the result of the phylogenetic analysis? What you find out? What was your finding? ये क्या बताता है? ये क्या बताता है? बताओ।
सर ये सिमिलरिटी फाइंड करता है सर, बताता है हाईएस्ट सिमिलरिटी।
तो क्या बताया आपको, क्या सिमिलरिटी किसमें क्या मिली बताइए।
किससे सिमिलरिटी दिखा रहे हैं योगेश्वरी?
सर और हीट मैप।
नहीं ये
किससे? नहीं बेटा किससे सिमिलरिटी दिखा रहे बताओ।
कंफ्यूज हो गई।
चलो कोई बात नहीं,
कोई बात।
और ये जो दूसरा है जो हीट मैप है, हीट मैप का जो कॉन्सेप्ट है, जो दूसरा आपका फिगर दिख रहा है, ये क्या बताना चाह रहा है? आपने एक्स एक्सिस में क्या लिया है, वाई एक्सिस में क्या लिया, ये कलर जो शो कर रहे हैं ये क्या बताना चाह रहा है, बता सकती हो
योगेश्वरी?
यस सर।
कलर क्या बता रहे हैं हीट मैप के?
चलो बेटा
कोई बात नहीं,
कोई दिक्कत नहीं, ये सर से आप डिस्कस करो और उसको बताओ है ना नेक्स्ट टाइम में।
ठीक,
वेरी गुड
Sir, you may please leave. Sony
Okay,
sir, please leave.
हाँ मैम ये
So I think it should be concluded.
Yes, sir. Actually, before the presentation, I had already started vote of thanks.
Yeah.
First of all, I would like to give special thank to the program convener as well as Edu, and special thank to the invited talk, Dr. Kamlesh Kumar Soni, and our session chairperson, Dr. Mahin Kumar Soni, who is head and professor of Department of Biotechnology, AKS University, and Department of Environmental Science, AKS University, SATNA. He is also DSW. He is very renowned and very helpful person, as well as a great researcher and scientist, and also involved in various types of social activities like NSS and they. I want to give a special thank to our session chairperson, Dr. Mahin Kumar Tiwari. Thank you so much. And the technical team of Edu Society, and a special thank to Dr. Aswini Bau ma'am, professor and head, AKS University, Department of Biotechnology, Satna. Thank you so much. Ma'am, anything you want
Thank
to
you,
speak?
Bharat sir, for great coordination of this session. Thank you, session chair, Dr. Mahin Tiwari sir.
Okay. Madam, I want to conclude the session from my side, sir.
Okay.
The session began with the invited talk, Dr. Kamlesh Soni, and his presentation provided the valuable insights into the molecular regulations of the tapetum-specific gene expression and its significance in the plant reproductive biology and genetic engineering approaches for the crop improvement. And the efforts of the Kamlesh Soni will be very useful for our students and the researchers also. Next, it is a very important that the students and researchers are also presenting their field work या project research, Ms. Srishti Soni, guided by the Dr. Sneh Wow, presented his work, and it was a nice presentation. I also extend my gratitude to the organizing committee, their excellent coordination, and for the providing the platform to promote knowledge sharing and collaboration toward the sustainable agriculture development. Overall, the session was highly interactive and intellectually stimulating. The presentation reflects the growing integration of the biotechnology and microbial research and computational approaches in the addressing agriculture sustainability and environmental challenges. I sincerely appreciate the efforts of the all presenters, their mentors, and active participation of the audience. And I also thankful the this program convener, Dr. Sneh Wow, and coordinator, and the Edu company CEO, Edu-Mentors Private Limited and Society to giving the chance to university and the departments to such a knowledge sharing platform. Thank you.
Thank you, sir. Ma'am, can we conclude this meeting?
Yes, sir. Aniket sir, can we conclude this meeting? Okay, sir. Conclude.
Yes, ma'am. We can conclude this.
Okay. Thank you.
Thank you, ma'am.